dear students. Okay, so our topic for today is dark reaction. As I have already explained that this process occurs in two phases. The first phase was light reaction, in which the products are oxygen, ATP, and NADPH2. Now the ATP and NADPH2, they are both called as reducing powers. The products like ATP and NADPH2, they are used here in this dark reaction for the conversion of for the reduction of this carbon dioxide into glucose molecule. So the actual motive for this dark reaction is the synthesis of glucose. So this dark reaction is also known as carbon fixation or Blackman's reaction or light independent reaction. As I earlier pointed out that this dark reaction is the name dark reaction is a misnomer, because this dark reaction, it can take place in presence of light also or in absence of light also. So this is actually called as light independent reaction, and this light independent reaction, it is supported by the products of light reaction, that is ATP and NADPH2. ATP is used for the energy, and this NADPH2 is used for the it acts as the reducing power. The next point about this dark reaction is that it takes place in the stroma of the chloroplast. Light reaction, it takes place on the thylakoid membranes, and this dark reaction or light independent reaction, it takes place in the stroma of the chloroplast. And this light independent reaction or dark reaction, it was first studied by Kelvin and Benson, and that is why it is also called as Kelvin cycle. And they studied this Kelvin cycle or C3 cycle in case of chlorella, an alga known as chlorella, single celled alga known as chlorella, and they got Nobel Prize in 1961 for this work. And this Kelvin cycle or Kelvin pathway, it occurs in all photosynthetic plants. Okay, and this C3 cycle, Kelvin cycle, is also known as C3 cycle, and it can be studied in different steps. The first step is carboxylation, where this carbon dioxide is reduced, it is fixed. Second is reduction, formation of succoses, and the third is regeneration of REBP. So here the first stable product of this cycle is PGA, that is known as phosphoglyceric acid, actually 3-phosphoglyceric acid. And this 3-phosphoglyceric is a C3 compound, carbon 3 compound, means it contains 3 carbons in its structure. That is why it is also known as C3 cycle. Here 1 RUBP, except RUBP means ribulose 1,5-bisphosphate. I will tell you this later on. This RUBP, 1 RUBP molecule, it accepts 1 carbon dioxide molecule at a time. So 6 turns of this cycle are required for the generation of 1 glucose molecule. Or and this cycle, it requires 18 ATP and 12 NADPH2 molecules are required for the synthesis of 1 glucose molecule. So both ATP and NADPH, they are obtained from the light reaction. And this C3 cycle occurs generally in temperate plants. Temperate plants means those plants where the climate is moderate, means there is not much higher temperature. Okay, so in case of Kashmir, we have temperate forests. Okay, and the optimum temperature is 10 to 25 degrees Celsius for this C3 cycle to occur. So here is the diagrammatic representation of C3 cycle. Here actually I'll explain it here. So this C3 cycle has 3 phases. One is fixation, where RUBP accepts carbon dioxide molecules and it forms 3-phosphoglycerate molecule. Second is reduction, where 3-phosphoglycerate, it plus 6 ATP plus 6 NADP, so it gives us 5 glyceraldehyde 3-phosphate molecules and 1 glyceraldehyde 3-phosphate molecule, glyceraldehyde 3-phosphate molecule, it goes to form a glucose or fructose molecule. And then there is the regeneration of REBP, 5 G3P molecules, glyceraldehyde 3-phosphate molecule plus 3 ATP, they gives us 3 REBP molecules. So here on the right side you will see the general cycle, this C3 cycle or Kelvin cycle. Now what you are seeing here is the first step is fixation of carbon dioxide. 3 carbon dioxide molecules, they are accepted by 3 RUBP, ribulose 1,5-bisphosphate molecules, to form 6 molecules of 3-phosphoglycerate. Now these 3-phosphoglycerate molecules, these accept 6 ATP molecules and 6 NADPH2 molecules. They are used here to form 6 molecules of glyceraldehyde 3-phosphate, and 1 molecule of glyceraldehyde 3-phosphate, it is used for the synthesis of glucose or fructose. Now remaining 5, they are used for the regeneration of RUBP. So this is how the cycle completes. Now here it is, in nutshell, the summary of the cycle is that it takes 6 turners to form 1 glucose molecule, and for that 3 ATP and 2 NADPH are required for 1 carbon dioxide molecule. So for 6 molecules of carbon dioxide to fix, 18 ATP and 12 NADPH2 are required. So in this box you will see that in means what goes in. What goes in is 6 carbon dioxide molecules, 18 ATP and 12 NADPH, and what comes out, what are the products, is 1 glucose molecule, 18 ADP and 12 NADP positive molecules. So this completes our C3 cycle. Tomorrow we are going to talk about this photosynthesis and factors of photosynthesis. Thank you.